How did muscular dystrophy blood testing become so advanced? Imagine a time when diagnosing muscular dystrophy relied mainly on examining muscles and observing symptoms. Today, thanks to incredible progress in genetic and molecular science, blood testing has become a powerful tool in identifying this complex group of diseases. Advances in technology have allowed scientists to analyze blood samples with remarkable precision, pinpointing the exact gene mutations responsible for different types of muscular dystrophy. This shift has transformed diagnosis from a mainly invasive process to a more accurate and less invasive one. Initially, doctors depended on muscle biopsies and electromyography to diagnose muscular dystrophy. These methods could be invasive, uncomfortable, and sometimes inconclusive. But as scientists uncovered the genetic roots of many muscular dystrophies, such as Duchenne muscular dystrophy caused by mutations in the dystrophin gene, the focus moved toward molecular testing using blood samples. Blood contains white blood cells that carry the same genetic information as muscle tissue, making it an ideal source for genetic analysis. The development of advanced genetic testing techniques played a huge role. Techniques like multiplex polymerase chain reaction and gene sequencing allow scientists to detect deletions, duplications, and small mutations in blood DNA. These methods can identify specific gene changes quickly and accurately, often within days. As genome sequencing became more affordable and widespread, it became possible to identify a broad range of mutations that cause different muscular dystrophies, making diagnosis faster and more precise. Research into how genetic defects cause muscle problems has also helped. For example, understanding how toxic RNA accumulates in myotonic dystrophy has led to the discovery of molecular markers in blood that reflected disease activity. Blood tests now can measure levels of certain proteins or enzymes, like creatine kinase, which indicates muscle damage. While elevated creatine kinase alone isn't enough for diagnosis, it supports genetic testing results and helps monitor disease progression. The progress in blood testing has also supported the development of gene therapies. Precise genetic diagnosis from blood samples helps identify the right candidates for treatment and allows doctors to track how well therapies are working. Additionally, innovative research uses blood cells reprogrammed into muscle precursor cells, called induced pluripotent stem cells, to study muscular dystrophy in the lab. This helps scientists understand mutations better and develop targeted treatments. Today, blood testing for muscular dystrophy offers many benefits. It can provide a definitive diagnosis without needing invasive muscle biopsies in many cases. It allows for carrier detection in females, early diagnosis in newborns and at-risk individuals, and personalized treatment plans based on specific gene mutations. Blood tests also help monitor how the disease progresses and how patients respond to emerging therapies. All these advances are thanks to the combined efforts of genetic research, molecular biology, and clinical studies. They have turned blood testing from a limited, indirect approach into an essential, precise tool for diagnosing and managing muscular dystrophy. These developments align with current medical guidelines and emphasize the importance of consulting healthcare professionals for personalized advice. Remember, each person's experience with muscular dystrophy is unique, and ongoing research continues to improve how we diagnose and treat this condition.